Chapter 10, Topic 2, Antimicrobial Resistance. In this topic, the learning objectives are to be able to discuss two possible ways that microbes acquire antimicrobial resistance, be able to list 5 cellular or structural mechanisms that microbes use to resist antimicrobials, and be able to discuss at least 2 novel antimicrobial strategies that are under investigation. We're going to be covering the development of resistance, types of resistance, new methods in antimicrobial therapy, and probiotics and prebiotics. We have talked about resistance and evolution throughout this course, so this slide should be a review of what we have talked about previously. As we mentioned, antimicrobials became available in the 1930s, and we began to see resistance as early as the 1940s. However, antimicrobials were not as widely used as they were in the later part of the century, and we began seeing an explosion of resistant microbes. This has led to a very important discussion on the overprescribing of antibiotics. Many environmental microbes carry natural resistance. This makes sense because they would naturally encounter other microbes producing these substances, and being immune to them wouldn't limit their environment. These environmental microbes can pass this resistance to pathogenic microbes through horizontal gene transfer if the cell doesn't naturally have this resistance. So just how do microbes fight off the antimicrobials? There are 5 key mechanisms. The first of these is to simply metabolize the micromolecules so that they are broken down and are no longer effective. Secondly, the microbe can evolve to have membrane receptors that don't allow the chemicals to pass into the cell. This is similar to evolution of viruses and host cells that we talked about earlier in this term. Third, if the cell can't modify the receptors, they can change the way the cell handles the chemical. This is usually handled by the cell pumping the chemical immediately out of the cell so it can't damage the cell. So if the cell can't prevent the drug from entering and can't pump it back out, the cell can then change the receptors within the cell so that the chemical can no longer act on its target. Lastly, even if the chemical can bind to its target and shut down the process, the cell can activate another pathway that can accomplish the same or similar events in the cell so it can continue functioning. So with all this resistant talk, we need to find new strategies to develop antimicrobials. You can see 4 of these types of strategies listed here. 2 of the most interesting strategies are using RNAi, or interfering RNA, to target the RNA within the cell. When bound to these, the RNA will be degraded and the proteins cannot be made. This allows us to target even more specific proteins within the bacterial cells. The other cool method is by using bacteriophages to target the bacterial cell specifically and leave the host cells alone. All of these are very exciting techniques, and we can only expect the strategies to expand as more research is performed. Lastly, we have talked a lot about your normal microbiota and how important it is for you. But what happens if your microbiota is lost during a round of antimicrobials? One option is to take probiotics. These are live organisms that we know are good for you and for your intestinal environment, and by eating these foods or taking these pills, it should help to restore your normal balance. A little more new to the market are prebiotics. These are nutrients that we know the microbes need to grow, and they can help encourage the establishment of new microbes within the gut. Overall, these are a huge area of recent research. As we have mentioned, the more we understand the importance of our microbiota, the bigger role of pro and prebiotics become in treating disorders. Well, that's it for this topic. Let's go over the learning objectives for this topic. Be able to discuss 2 possible ways that microbes acquire antimicrobial resistance. Be able to list 5 cellular or structural mechanisms that microbes use to resist antimicrobials. And be able to discuss at least 2 novel antimicrobial strategies that are under investigation.